All right, hello everyone, and welcome to today's cryo-EM Current Practices webinar. You're joining the 3 national centers established by the NIH Common Fund Transformative High-Resolution Cryo-Electron Microscopy Program. My name is Christina Zamani. I'm a scientist and training liaison at the National Center for Cryo-EM Access and Training, NCCAT. And with me here today are my colleague at NCCAT, Ed Eng, joining from the Stanford SLAC Cryo-EM Center, S2C2, is Michael Schmidt, and from the Pacific Northwest Cryo-EM Center, PNCC, is Jeanette Myers. Jeanette will introduce today's speaker, Hanbin Jeong, after my colleagues from each of the centers give a quick update on the resources that we make available at no cost to the research community. This webinar is a regular event we host the last Thursday of every month, and we highlight methods, tips, and tricks that groups are using to obtain and interpret the data they can collect using National Cryo-EM Center resources. So we will have a talk on February 23rd. We're waiting for final confirmation on our speaker. So for now, it's a mystery talk, but look out for announcements for that talk soon. Also, if you missed our December talk, in place of a formal seminar, we hosted an informal roundtable on the topic of particle picking. And so if that's something of interest to you, you should go find the recording along with those of our past talks on our YouTube page or the events page at cryoemcenters.org. And if you like this roundtable format and have topics of interest for future roundtables, please reach out and let us know. What we want to discuss are the topics that are of greatest interest to you. So let us know. Final logistics from me. On Zoom, we're going to be watching the Q&A box for questions. So please use that Q&A and not the chat to send us questions. Jeanette will be moderating questions at the end of the talk. And that's it for logistics from me. So I'm going to turn over to Mike, who will let us know what's going on at S2C2.
Hi, I'm Mike Schmidt. I'm one of the co-directors of the Stanford SLAC Cryo-EM Center, S2C2. We have Krios's and Talos available for training and for high-resolution data collection and for exploratory searches, i.e. screening. We are doing all of those. We can do all of those on site now or remotely. Users either send us the grids or come and make grids on site. Our training and our exploratory activities are going well, and we hope to see you have a look at us.
Thank you, Mike. I'll turn over to Ed.
Hello, I'm Ed, the manager of NCCAT here in New York. We have 4 dedicated Krioses for high-end data collection, a robust grid preparation screening service with applications for chameleon access, a blot-free vitrification device, pardon me, and a robust cross-training program. Something that is happening on February 8th, I'll be at Ask the Expert with Therma Fisher about cryo-EM national centers and how we're a go-to resource for getting started in cryo-EM. I'll be joined by Becky Thompson because cryo-EM is not just a national effort, but a global effort. And lastly, if you're at the Biophysics Society, all 3 national centers will be at booth 701. So learn more at nccat.nyspc.org.
Thank you, Ed. And finally, I'll turn over to Jeanette.
Yeah, hi, I'm Jeanette. I'm a microscopist at PNCC. And we have 2 different types of proposals. One of those is our screening access, where you can apply to come on site for up to 5 days to just make grids and screen them with staff. And that's for people that might not be able to freeze grids otherwise. And then we have our general access, which just gives you 480 hours of microscope time a year over 2 years, which we can do screening or data collection at that time. And if you have a general access proposal, you can also come to our training workshops and things of that nature, which cover sample preparation through data processing. And your proposal will also be assigned a scientific point of contact, or SPOC, which is my position, who can answer your questions about sample optimization, data collection strategies, etc. And we have one Arctica, 4 Krios, all have K3 cameras, we have a Falcon 4i, 3 energy filters, and we have a Vitrobot and like a GP2 for sample prep. So hopefully we'll hear from some more of you guys soon. And so with that, I'll go ahead and introduce Hanbin. So Hanbin received his doctoral degree from Ulsan National Institute of Science and Technology in South Korea. And his doctoral research mainly focused on structural studies of membrane contact site protein complexes using X-ray crystallography. He also studied non-vesicular lipid trafficking between ER and mitochondria. And Hanbin joined the Gouaux lab in 2021 to study the composition and molecular organization of the mechanosensory transduction channel using cryo-EM. And today he's going to tell us about their recent work on the TMC1 complex. So with that, the floor is yours.
Yep, thank you for nice introduction, Janet, and thank you for invitation. Hello, everyone. This is Hanbin from Eric Gouaux in OHSU. Today, I'm going to talk about the structure of TMC1 complex, which is involved in mechanosensory transduction. So there are 5 senses of how our brain watch in the external world. The sensory systems all have the job of taking each stimuli from external world and converting those into electrochemical signals. The eye is converting visual stimuli into electrochemical signal, and the tongue's tasting molecules converted taste into electrochemical signal, and the ear and the nose too. And this is a major focus on neuroscience, trying to understand the genes and proteins involved and trying to elucidate the mechanism of how this information converted into electrochemical signals. And during the last 50 years or more, we made some important progress in the case of vision, smell, taste, and touch, just last year. And interestingly, the auditory system has not been recognized in this way because the auditory system is very delicate, complex, and it has a very challenging system to understand each mechanism. So our lab have been focused trying to understand the ear and what the molecules are convert sound information into electrochemical signals. And these all begin with the sensory helices. The sense of hearing occurs through the cochlear in inner ear, which have specialized receptor called helices. And there are stereocilia at the top of the helices, and each stereocilia is connected with the tip link structure. So the mechanical force, which deflect the stereocilia to the one side, result in the tension on the tip link, so that the tension makes open the TMC1 ion channel, which is closely located to the tip link structure. So if you look at this complex more closely, the tip link structure is connected with the TMC1 mat channel complex, and when the stimuli is applied, the force from tip link induce open the channel, and the cations such as calcium and potassium ions on the extracellular fluid come into health cell and generating electrochemical signal. And This figure shows the electron microscope image of tip link from guinea pig cochlea, and you can see the connection clearly in between two cilia. And there has been a lot of progress in the last few decades in terms of identifying match-channel component proteins such as proto-cadherin 15 and cadherin 23, which make tip link, and cadherin 23 is connected with cytoskeleton proteins, and there are several proteins consisting of MET complex, for example, TMIE, LHFPL5, CYP2, and the ion channel protein TMC1 and 2. And we now know that the proto-cadherin 15 and cadherin 23 make tip link structure with their multiple cadherin domain. And we also know the binding interface. And about 5 years ago, postdoc in Gouaux, Jinpeng and Johannes elucidate the structure of LHFPL5 and proto-cadherin 15, which shown that the proto-cadherin 15 and LHFPL5 form a heterotetrameric complex. And it shows that the extracellular flexibility of each complex on the membrane, and it also gives a clue that how the force from tip link may be transduced into the stereociliar membrane. So, okay, then the remaining question is how this tip link structure is connected with the match channel complex, and what is the mechanism of gating this channel? However, despite of several decades of investigation, the molecular mechanism of TMC1 complex and the structure of TMC1 complex is still unknown. We had tried to put a lot of effort on developing the overexpression system. However, mostly it failed because, as you can see, one of the reasons is the overexpressed TMC1 just trapped in the ER, and they never reach the plasma membrane. So they are somehow not properly folded in the overexpression systems. So we tried alternate way, which planned to isolate native protein from animal source. We used to isolate the specific protein from animal using high affinity antibodies, and those techniques are one of the strengths of Gouaux. And the advantage of isolating native protein from living source is we could preserve each native environment, such as auxiliary proteins, post-translation modification, and the lipid molecules, which help to stabilize protein structure. And also developing a cryo-electron microscopy technique enabled to analyze the protein structure with a very little number of proteins. However, in order to isolate native proteins, we need strong antibodies to pull down the target protein from animal source. But we don't have such strong antibodies for isolating TMC1 proteins or each component proteins. We could use the transgenic mouse model, introducing the affinity tag on each target proteins. For example, the native Casper channel studies from mouse sperm. However, the isolating TMC1 protein from native source is kind of also challenging because there are very small number of TMC1 channel complex in cochlea. compared to photoreceptor in visual system. So we look for another way and finally decide to use C. elegans as a model. Many scientists already have shown that C. elegans have all of the components we have in the vestibular system, and each TMC1 involved in the mechanotransduction of worm, like touch avoidance. And most importantly, we can grow a large number of worms for native structure studies. So we made transgenic worm line, which has fluorescence tag and affinity tag on the end of TMC1 genes so that it can express TMC1 endogenously. And this is a head neuron of transgenic worm. And as you can see, it expressed well. And to grow a large number of worms, we used liquid culture system. And we use bacteria as food source of worm. And initially we use this NGM plate, agar plate, for growing a large number of worm. And after that, we transfer worm to the flask and do liquid culture more to produce a large quantity of C. elegans. And finally, as a result, we could get around 60 millions of worm from the culture, which is around 40 to 45 million volume of worms. So here is each detailed step for isolating TMC1 from C. elegans. So first, to lysis the worm, we grind the worm. It sounds a little horrible, but yes, we use a bore mill system to disrupt worm and to make the fine powder in the liquid nitrogen environment. And it is actually not usual method to lysis a sample. We used to use sonication, French press, or homogenizer to break down the tissue or cell. However, this bore mill method is efficient to break apart the worm membrane tissues and help to stabilize the oligomeric stage of TMC1 complex. And after then, the worm powder was solubilized in the presence of GDN detergent. And because we knew that there are a little amount of TMC1 protein in worm, we needed to find efficient way to isolate much TMC1 protein during the solubilization step. And I used the fluorescence detection stage exclusion chromatography AFSEC system to screen diverse pH conditions and salt concentration. And a kind of interesting thing is when I changed the high pH, such as pH 9, the solubilized TMC1 increased more than 3 times compared to pH 7 condition. And moreover, when I changed the salt concentration to 50 millimole, which is lower than normal TBS, usually 150 millimole, the isolation efficiency also increased more than twice. So I set the solubilization buffer with high pH and low salt condition with the detergent GDN. So after solubilization, centrifuge the sample and the supernatant was applied to anti-flag affinity agent, and TMC1 proteins was eluted by incubating 3C protease and concentrated and loaded onto stage exclusion chromatography column. And I collect the putative dimeric peak and concentrated to make cryo-EM grid, as well as mass spec analysis. So we could get around 100 nanogram of protein from 60 million ones, and the estimate molecular weight is around 800 kilodalton, which means it has multiple TMC1, and perhaps it forms complex with other auxiliary proteins. So we did mass spec analysis, and it identified TMC1 and 3 other proteins, CAM1, TMIE, and ARRD6. And CAM1 is orthologue of mammalian CYP2, and CYP2 and TMIE known as binding partner of TMC1. And interestingly, there is an arrestin domain protein which haven't been described as binding partner before. So yeah, as I mentioned, the total protein I got is less than 100 nanograms. So for TMC1 project, I couldn't use the holy grid because I can't make such high concentration protein sample. So I decided to use the grid with 2 nanometer carbon support because I can apply relatively low concentration of protein compared to the holy grid. So this micrograph shows the initial result from cryos, and I could observe putative some protein particles on the grid, and this figure shows the 2D classification result. And I also could see some feature of particles on 2D classification, like this side view. However, there are some orientation issues that many particles shows top and down views here. And also, it is hard to observe the transmembrane helices in detergent mice. So finally, I could get around 8 angstrom resolution of map, which is hard to see the feature of transmembrane residues. So to overcome these particle orientation issues, I tried to run flow discharge in the presence of amylamine. It is known that the flow discharge method used for cleaning the carbon surface as well as modifying the carbon surface. So unlike the normal flow discharge with air, the amylamine changed the surface charge to the positive charge. So it is used for observing negatively charged biomolecules such as DNA, and there are some evidence that treating amylamine improved the orientation problem, for example, like the 20S proteasome complex presented in here. So I saw the filter paper with amylamine and place it in a flow discharge chamber and run flow discharge. And the result looks promising that, as you can see, this 2D classification with and without amylamine, I could observe much more side view in the presence of amylamine. And moreover, now I can see clearly the feature of each transmembrane helix and compare to the air flow discharge method. And as you can see, these two direction distribution figures some orientation issues somehow get better after I use amylamine glow discharge. And using this amylamine also helps somehow eyes thickness of sample, and I think the eyes on the amylamine grid tends to thinner than normal condition that improve the contrast of micrographs. And so finally allow me to solve the high resolution structure of TMC1 complex in the end. So this figure shows the overall data processing strategies, and I collect around 26K movies in 5 days, and motion correction and CTF estimations were performed by CryoSpark, and I did block picking to pick particles, and after one round of 2D classification, junk particles were removed and resulting in 3.2 million particles in total. And using the model from ab initio, the heterogeneous refinement was performed. And in this step, the probable monomer TMC1 complex and the detergent micelle and some junk particles were removed. So after that, 1.6 million particles were re-extracted from unbinned images. And then heterogeneous refinement using the C1 symmetry was performed with 8 classes, and among them, these 3 good classes were selected. And after one round of heterogeneous refinement using the C2 symmetry, the 2 classes were distinguished here. And one more round of heterogeneous refinement was performed to sort out homogeneous particles. And finally, non-uniform refinement was performed, including defocus and CTF refinement. So yeah, we call this model in expanded form, and the same strategies were applied to the other conformation, and we called it the contracted form. And among the initial 8 classes from heterogeneous refinement, one of the class had an additional density feature in here. So further heterogeneous refinement and 3D classification without alignment was performed in Reliant. And after one more round of heterogeneous refinement in CryoSpark, we finally get homogeneous particles with this feature. And to improve the extra density, we did local refinement with mask, covering the extra density on a cytosolic basis. So as a result, I could solve 3 different structure form of TMC1 complex, and this is an example of micrographs I collect from EM and representative 2D class average image of each conformation. And as you can see, I found 2 different conformations of TMC1 complex, expanded and contracted conformation, and the complex with arrestin protein, which adapt the expanded conformation. And because the expanded conformation has a few more density feature than the contracted form, I will focus on the expanded conformation after now. So this is the overall architecture of the TMC1 complex. And as you can see, it is dimeric form, and it has 2-fold axis of rotation at the center of 2 TMC1 subunit. And CAM1 cover the cytosolic helices of TMC1, and TMIE proteins located each edge of the complex, the peripheral side. And interestingly, the last transmembrane helices, TM10 of TMC1, form a domain swapped structure in this complex. And yeah, these movies help you to understand the overall structure of the complex. And as I mentioned, the last transmembrane helices located here, domain swapped manner, and the TMIEs and common proteins are here, red and yellow respectively. And these small red spheres are calcium ions on EF-handed motif on CAM1 and TMC1's pore regions. And these black ball and stick are lipid or detergent-like molecule in the structure. And I also found the glycosylation site, glycosylation of asparagine here in green. So according to structure, the complex has 2 copies of TMIE, and the binding interface between TMC1 and TMIE consists of not only protein-protein interaction, but also lipid-mediated interaction, such as phospholipid and permethylation of cysteine. And interestingly, there is a large cavity between transmembrane helices of TMIE and TMC1, and there is where ordered phospholipid which is coupling TMIE through the pore-forming helices of TMC1. And moreover, the permethylated cysteine extended along these helices. So it suggests that the TMIE protein is involved in the gating TMC1 by sensing the lipid membrane changes. And there are common proteins on the cytosolic faces of TMC1, which has 3 EF-handed motif, and it covers the cytosolic faces surface of TMC1, and almost every cytosolic helices of TMC1 are interacted with this common protein. And there are calcium ions on the last 2 EF-hand motifs, and we don't know the exact role of calcium binding to CAM1. But according to each close location to the TMC1 pore, we guess it might modulate the ion channel function. And one interesting thing is, during the particle classification, we found a small number of particles which has arrestin domain protein bound to CAM1. And we do not know why the only one arrestin bound to CAM1, and we speculate that one subunit unbound from the complex or just partially occupied due to the weak interaction. And further studies are required to address these issues, as well as the function of arrestin protein in C. elegans TMC1 complex. And based on the structure we served, each TMC1 has ion permeable pathways, and these look closed. We could use the same protein fold families such as TMM16 and OSCA to guess where the ion permeable pathway of TMC1. And it is not highly polar pathway, and actually it has a number of acidic residues along the pathway, which is inconsistent to being a canonical calcium channel. the channel we have our ear is basically cation non-selective channel, including calcium ion or potassium ions, and we don't know really anything about this channel. But just from the pore lining architecture here, I would say it is likely to be a cation channel. And yeah, this figure shows the multiple point mutations of each protein linked to deafness and hearing loss in human compared to C. elegans TMC1 complex structure. And much of them are located to pore lining architecture area of TMC1 and also a binding interface between TMC1 and TMIE, as well as the CAM1, which means those mutations cause defect on ion conducting pathway and destabilized protein structure. And one thing I want to show you is the simulation work from our collaborator. The EMAD group put the complex structure into lipid bilayer, and interestingly, the amphibatic H3 helix on TMC1 insert deeply to the membrane and almost parallel to the membrane. And it caused the membrane bilayer thin. And this group of 3 helices are pretty close to the pore of TMC1. So just wild speculation, if the membrane thin, these helices rise, move up and down like float, and this may involve in gating mechanism of TMC1 complex. And these results are supported by the fact that many mechanically activated ion channels, for example, Piezo, Bosca, and Dong-Z, seems to share a common feature of these amphibatic helices. And these amphibatic helices are known to sense or induce membrane curvature and closely relate to the gating pore structure. So we speculate that our H3 amphibatic helices may also involve in gating mechanism of TMC1's pore. And one last thing I want to show you is, as I told before, during the particle classification of cryo-EM processing step, I found 2 different conformations of TMC1 complex, the expanded and contracted conformation. And we superimposed 2 models, and the result shows that the axis of rotation is almost parallel to the membrane. And this movie shows the conformational changes between 2 conformations. And as you can see, the movement within 2 conformations is breathing-like motion. And we also named accordion-like motion. And the axis of rotation is parallel to the membrane. So I believe these 2 conformations suggest the conformational flexibility of the TMC1 complex. And it also shows the possibility that the deformations of membrane may induce the conformational changes of TMC1 complex. And these are kind of interesting because when you compare these 2 models on MD simulation, these 2 conformations generate distinct membrane deformation patterns. And as you can see, the expanded conformation induce more strong membrane thinning or thickening, close to the protein. And as a result, it makes a longer range of membrane deformation pattern, which is around 50 angstrom away from the protein. However, in the contracted conformation, it does not make such membrane deformation patterns here. So, yeah, I think what we have for sure is it could be wrong, but based on the sequence, the C. elegans proteins are highly conserved to the sequence of mammalian. So I would say this is likely kind of the core complex structure of transduction channel. And there are multiple ways to which force bind to couple the gating mechanism. Maybe something binds to TMIE and directly pull the channel to open the gate, or indirectly gating the channel by thinning the membrane with generating hydrophobic mismatch. Yeah, we just don't know. And further studies will build on this work by elucidating the open channel conformation of the TMC1 complex. And yes, of course, the biggest question is how these all resemble in mammalian, where is protocatecholine 15 at HPL5, this TMIE structure, and do they interact directly to the TMC1 complex or indirectly? Yeah, we don't know for sure. And yeah, there are interesting things and a lot of fun to be elucidated here. So yeah, I have one more slide here. And thanks Eric and all Gouaux members and Congress lab members, especially Sarah and April, for a lot of discussion and initial setup for warm culture system, and EMAT group for MD simulation, and Ashok for mass spec analysis, and thank PNCC and John earlier for screening and collecting cryo-EM data. Yeah, thank you all for your attention.
Thank you, Hanbin. That was absolutely gorgeous. And just like me, a few people in the audience have questions, so I'll let Jeanette take over and moderate some questions.
Sure. Yeah, I'll just go through them in the order that they were received. So first one is very interesting talk. Could you expand on how you set up your amylamine glow discharge strategy?
Yeah. Yeah, I think you need a separate machine to prevent cross-contamination, because once you learn amylamine on this glow discharge, you never use this as the normal condition. And I soak amylamine into filter paper, usually 50 to 100 microliters of amylamine. And yeah, just put the filter paper on next to the slide glass and learn glow discharge. That's it, yeah.
Okay.
Mm-hmm.
We had a comment, I'll add that it was a very interesting way to solve the orientation problem with changing the glow discharge. I agree, it was pretty cool. So mark that one off. Okay, next one is, besides the evidence that TMC, CAB2, LHP15, and TMIE are part of the MET complex in mammalian hair cells, what is the direct evidence to suggest that TMC is indeed MET channel in mammals?
So the question is, what is the evidence that TMC1 is ion channel or MET channel?
In mammals, yeah, I think. Feel free to send a clarification if I'm getting that wrong, anonymous attendee.
Yeah, I think many studies have already shown that the TMC1 and maybe TMC2 is ion channel or channel complex and involved in hearing and balance in vestibular system.
Okay, let's see. Next one is, just out of curiosity, have you thought about using the tagged protein without C3 cleavage for grid prep? In my experience, some tags actually help with the orientation problem.
Actually, yes. And in the beginning, I just use just 2x flag peptide to elute the TMC1 from beach. But yeah, the main problem was the purity of protein was really bad, and I think that's because I use a pretty large amount of flag beach, like 300 or 400 microliters of beach, and the amount of TMC1 from worm is less than 100 nanograms, so some non-specific binding proteins also co-elute with 2x flag peptide. So yeah, the purity was really bad. So I changed to the method to use 3C protease to elute TMC1 from the beach.
Gotcha. Yeah. Well, could be a good tip for someone out there having some orientation issues that have more protein. Okay, next one. What was the final concentration of the protein that you put on the grid?
Yeah, actually, I think it's less than 10 nanogram per microliter.
Yeah.
It's hard to measure the exact concentration.
Yeah, not much though. Okay, and then this is a multi-parter. So the next one is, why do you think only one copy of ARD6 is attached? And
Yeah.
have you seen particles with both sides occupied? And then this is all kind of one, I'll keep going. Does the mass spec quantification suggest a conformation with 2 copies?
Yeah, as I mentioned, I think one subunit unbound from the complex or just partially occupied due to weak interaction. And yeah, what is the second question? The mass spec?
Yeah, they asked, have you seen particles with both sides occupied, and if the mass spec suggested that there was a conformation with 2 copies?
Oh, yeah, I think we didn't find out the like the number of stoichiometry of each proteins on mass spec. Yeah.
Okay. Well, let's see the next one. We have a question. Just to clarify why you chose amylamine, was there something maybe else you considered before? Or is that one of the first things you tried? Or
yeah, it is the first thing I try. After the air glow discharge not work for me. Yeah.
I assume part of the reason you picked it is because it helped with the changing the particle orientation.
Yeah,
right.
I think it not only changed the particle orientation, but also improved the contrast of particles. And I think, as I mentioned, that the treating amylamine tends to thinner the eye thickness, so that it helped to improve the contrast of particles.
Yeah. Okay, that was a nice bonus. Let's see. And then have a question. What is the significance of being a dimer? I presume TMC1 being a dimer.
Well,
Or we can send a clarification on that too, if that's not clear enough.
Actually, yeah, I had a similar question. Like, is there any interaction within that dimer interface that seems like it could be relevant to gating at all, or does it just happen to be a dimer for maybe other reasons?
yeah, I think this dimerization is induced by the domain swap feature of TM10. And I think, I'm not sure, but I think maybe this domain swap feature may hold the dimeric complex onto membrane. So maybe, yeah, I'm not sure.
Okay, and then we have a question here. If you could, they'd like a little more information on your data processing, like when do you use heterogeneous refinement over homogeneous refinement? This is from someone who's pretty new to the field, they specify. Maybe like, how do you make your decisions? Yeah, like when to do different types of refinement and things like that. If you have like a general comment on that.
Yeah,
If you can.
actually, it is my first project of cryo-EM, using cryo-EM, and first time to use CryoSpark or Reliance. So many other lab members helped me to success this project. So yes, I think each is kind of, I think it just follow the like protocols, follow the CryoSpark strategies, I think. Yeah.
Well, actually, can I ask, so you show this nice workflow, but like how many other jobs did you try that like aren't included here, right? Like at each, like if you actually drew how many jobs you did that you then like didn't carry forward or abandon, like how much wider would this slide be?
Yeah, a lot. Yeah.
Yeah, I don't think we can give like a very succinct answer to that one, but let's see. Done. Okay, next one. Nice talk. I might have missed this. How did you decide which proteins do you think are the subunits of your complex according to your mass spec result? Like how you chose the first 3 proteins. Why didn't you choose the 4th one on your list? How do you set up the threshold?
Oh, actually, yeah. Actually, I analyzed 2 samples. One is TMC1 isolate from worm, and the other is just control samples, which use just wild type worm and follow the same protocols like isolate TMC1 and elute nothing from the beach and send the sample also to the mass spec. And I just compare each TMC1 and the positive negative control. And as you can see, the TMC1 and other proteins hit, but the negative control just 0 here.
Okay. And then we have a question here. Would you explain a bit more clearly about how you introduced the fluorophore to your native protein?
Oh, I ordered one to the C. elegans company. Yeah.
Oh, okay.
Yeah.
Easy enough.
Yeah, and actually, if I can ask a related question, do you do any fluorescence imaging on like the whole worm before you start your prep? Or are you only starting to use the fluorescence at the FSEC stage?
Oh, I did before this, yeah, before the isolating TMC1. Yeah. to see they are where translocation in their place. Yeah.
And then we have a question here. May you please reiterate the significance of amphipathic helices in mechanosensitive ion channels? How do they affect the orientation or manner in which they incorporate into the membranes? So, that's just if you have any more to say about that.
Yes, it is common feature that the mechanosensory ion channels have this kind of amphipathic helices, and yeah, we also see that the H3 helices or KMC1 is kind of amphipathic feature, and actually it inserted into membrane by MD simulation. So yeah, we just expect that they might work as other amphipathic helices in other mechanosensitive ion channel, and yeah, they use to like sensing membrane, and sometimes these are involved in gating ion channels by, I'm not sure, but it kind of holding the position on membrane and like move or the position. Yeah, I don't know exactly the law, exactly function of each amphipathic helices in the mechanosensitive ion channel. Yeah.
Let's see. And then we have one here. Is amylamine a standard go-to additive for helping with ice thickness and particle orientation? I could say, I think it's known that it helped with the particle orientation, at least for the example that he showed with negative staining. I haven't personally used it, but we also can't really dedicate an entire setup just to doing that, like he said you need to do. So, if you have anything else to say.
Actually, I'll point out, thank you to the anonymous attendee who actually tossed up a link to a paper. If some of you have more questions about amylamine, there's a reference in the Q&A you can go grab. So thank you for sharing. We appreciate that.
Yeah, thanks. I'll leave that one up so hopefully people can grab it if they want it. Let's see. Here's one nice talk. Have you tried other methodologies besides amylamine to solve the particle orientation issue? It seemed to me like that was the first thing you tried and it just kind of worked, but if that's not correct, let us know.
Yeah, actually I tried other, like other using graphene grid, or it is known that using magnesium acetate, applied magnesium acetate on grid also changed the feature of the carbon membrane, but yeah, it's not work for me.
Okay, and one more here. Did you directly test the benefit of liquid nitrogen cell disruption compared to other methods such as sonication or French press for this project?
Yes.
Did it help preserving the structure, increasing the yield?
Yes, I did, and yeah, I think we used to use sonication to break apart cell and membrane, but sometimes it feels that the sonication power is too strong to break the complex apart. So yes, I did compare between the Boromir method and sonication and homogenizer and French press, and yeah, the Boromir method is the best to preserve the oligomeric stage of TMC1 complex. Yeah.
Yeah, so we hit the hour, and I'm guessing a number of people have to run off to other meetings or their lab bench and try out some new techniques based on what they heard today. So thank you everyone for joining us. Those of you embarking on your own cryo-EM journeys, we wish you a lot of luck, and the national centers are here to help you. So please let us know how we can help, and come join us again for future talks. And thanks again to Hanbin. That was absolutely gorgeous and some really helpful tips and tricks. So thank you.
Yeah, thank you all. Bye.